If you're a human, chances are that you've been bitten by a mosquito before. These bites aren't just annoying, they can be lethal. Mosquitoes kill more than 1 million humans every year, more than any other creature. But a recent breakthrough stands to change that. With a genetic engineering technology called gene drive, we can disarm mosquitoes for good. A genome is just like an instruction manual for building an organism. It's written in the language of DNA and it has genes as the steps. Through selective breeding, humans have been changing the genomes of other organisms for a long time, like how we bred dogs from wolves. However, selective breeding cannot target specific genes, and it takes forever. But what if you could find and replace only the malaria-enabling genes? CRISPR is a gene editing tool that does just that. CRISPR relies on a protein called Cas9 to cut DNA. We tell Cas9 what to look for and then insert Cas9 into the mosquito. Cas9 will find the gene and cut it out. If we insert a new gene, the cell will rebuild the DNA using this new gene. But this technique doesn't completely solve our problem. When a modified mosquito mates with a wild mosquito, only half of their offspring will have the desired trait. With each generation, the modified trait gets diluted, effectively disappearing. What if we could ensure that the modified trait was always passed on? Then, every mosquito on Earth would eventually be malaria resistant, the perfect solution to our problem. How would we do this? We would need to propagate Cas9 in addition to the malaria resistance gene. Well, Cas9 is a protein, so we can make a gene that tells cells to produce it. Let's call this the Cas gene. Then we can introduce both the Cas gene and the resistance gene to create modified mosquito 2.0. When this mosquito mates with a wild mosquito, their offspring would inherit the Cas and the resistance genes from the modified parent, and the malaria-enabling genes from the wild parent. Cas9 produced from the Cas gene will cut out the malaria gene in the offspring. Then, the cell will repair the break by copying both the Cas and resistance genes. Because of this, all the offspring would have only the modified version of the gene, gaining malaria resistance and the ability to propagate it. This is a CRISPR gene drive, and it might be our best bet against malaria. Gene drives occur in nature too, and are a phenomenon where specific genes are driven through a population, at a higher probability than allowed by natural selection. Usually, genes are passed on only if they help the survival of a species, but gene drives spread genes even if they don't help the species at all. But with such powerful technology, we have a lot of questions to answer. Gene drives might have unexpected outcomes. With the case of malaria, we're changing a very small part of the genome, so we can expect mosquitoes to still look the same. Still, there are many important ethical issues we need to consider. As something that has the potential to impact the public, it's crucial for everyone to be involved in this debate, including you.